So move on to our second speaker. So our second speaker is Dr. Caroline Perner. She got her MD degree from Otto von Guericke University in Magdeburg, Germany. After finishing her neurology residency, she came to the US. She joined Dr. Caroline Sokol's group as a postdoc in the Center for Immunology and Inflammatory Diseases at MGH. In the coming spring, Caroline will move back to Germany to start her own research lab at the University of Greifswald. So she will continue studying neuroimmune interactions as an independent PI. So Caroline, the floor is yours.
Thank you for the introduction, and I share my screen. I hope you can all see it. Yeah, so thank you also for the organizers and for the invitation.
It's still the presenter mode. Yeah,
that's the presenter mode?
Yeah.
Okay, do you see it now?
Yep, it's perfect.
Yeah, good, perfect. Yeah, so yeah, I'm very happy to present a protocol for the dissection and culture of dorsal root ganglia that we published in the STAR Protocols journal this year. So the image that you can see on this slide is a dorsal root ganglion that was dissected by me using exactly this protocol and was imaged by our collaborator, Dr. Birgit Perner from the Leibniz Institute in Jena in Germany using whole mount light sheet microscopy. And as you can very nicely appreciate, the pseudo-unipolar anatomy of the sensory neurons with their cell bodies located within the dorsal root ganglia, while the axon travels without any interruption from the periphery to the spinal cord, allowing for a very fast and undisturbed action potential. So in the green color, we stained, in this case, for touch 1, which would stain neurons, while in red, we stained for CD45 positive immune cells that you can see probably located mostly in the capsule in this case, and tissue surrounding the sensory neuron soma. So first I want to give like a brief overview about the anatomical location and also the composition of the dorsal root ganglia by referring to human anatomy. There are just not that many nice graphics about mouse anatomy in this case, and most of the research is anyway translational research, I guess, so I think this is doing the purpose good. So dorsal root ganglia are part of the peripheral nervous system. They are located within the intervertebral foramen, very close to the spinal cord. And they are separated through the meninges. The dorsal root ganglia are the location of the cell bodies for only the peripheral sensory neurons, including visceral afferent neurons and somatoafferent neurons. And these neurons can sense proprioception, so basically how you're, like in which position your arm or your legs are, or the joints are, or the muscles, light or rough touch, vibration, temperature, pain, and itch. So everything that gets sensed, like basically these neurons have their cell body within the dorsal root ganglia. There are no cell bodies of any sort of efferent fibers, like somatoefferent fibers that are for like muscle innervation, or visceral efferent fibers like belonging to the autonomic nervous system, like the parasympathetic or the sympathetic system. These neurons don't have their cell bodies within the dorsal root ganglia, but within the spinal cord in the gray matter. Besides the sensory neuron cell bodies, the dorsal root ganglia are composed of satellite glia cells that are surrounding the soma, like the cell bodies of the sensory neurons that you can see here, like with, of course, fibrocytes that build the capsule, And you also have some immune cells like macrophages, also T cells, lymphocytes around in the dorsal root ganglia. Like in contrast to the central nervous system, the dorsal root ganglia do not have a tight vascular barrier like the blood-brain barrier, but are supplied through fenestrated capillaries. This dissection protocol for dorsal root ganglia that I will present, it can be used to harvest dorsal root ganglia for microscopy and histology, as I showed in the first slide, but also for the isolation of sensory neurons for in vitro experiments, including cell culture. And in this protocol, we are describing sensory neuron culture for the purpose of neurotransmitter and neuropeptide release. But these cultured sensory neurons, or like the basically freshly isolated DRGs, could also be used for applications like electrophysiology and calcium imaging, as well as for any kinds of omics research. The protocol that I'm presenting can be divided in basically also 4 steps that include the perfusion of the mouse and the dissection of the spinal column in step 1, the picking of the dorsal root ganglia by using a dissection microscope in step 2, the dorsal root ganglia digest and culture in step 3, and as an example for a downstream application, the stimulation of the sensory neurons for neuropeptide release in step 4. So the STAR protocols paper therefore include all the protocols also for all necessary media and buffers that are necessary for the whole protocol. And they should be prepared best before you start. So especially for the sensory neuron culture applications, we recommend also coating a tissue culture plate with laminin on the same day before you start the harvest and digest of the DRG. And as you can see, like most of the media that we are using for this protocol are pretty simple. Like we have like some supplemented DMEM with some FBS, some penicillin. We have supplemented neurobasal media, which is probably the most complicated one, which we basically prepare like sterile, like supplemented like this, which is like the basic. And then like if you culture, we just add on the day on top of this before the culture some cytokines. And this is the enzyme mix with collagenase displase that we need for the digestion. So the first step is the perfusion of the mouse with phosphate buffered saline. The purpose of this procedure is to exchange the blood with the buffer to reduce blood cells in your samples. And to better understand this procedure, I quickly want to explain the heart anatomy and function. So the venous deoxygenated blood enters the heart through the right atrium and then goes to the right ventricle, where it then basically gets pumped into the lungs to get oxygenated. And once it's oxygenated, it enters again through the left atrium to the left ventricle, where it gets pumped out and distributed via the aorta into the whole body. And what we do when we do the perfusion is we cut into the right atrium so the buffer and the blood can escape. And then we pierce the heart and at the left apex or in the left ventricle and perfuse with 2 to 4 ml room temperature PBS, so that basically the PBS distributes very nicely via the aorta in the whole body, and the blood and the buffer can escape via the right atrium. So you can confirm a proper perfusion by appreciating a color change in blood-heavy organs like the liver that will turn from a dark red to a more beige brownish color. And maybe one more thing, if you are planning to performing histology or microscopy with the DRG, we recommend using already 4% paraformaldehyde in your PBS for the perfusion. So for the dissection of the spinal column, you can turn the mouse then to the ventral side and lift the skin at the base of the tail and make an incision over the whole back to the neck. And then you can pin or cut away the skin to the side, so you have like good access basically to the back of the mouse. Then the first thing is that you make a cut with your scissors at the lumbosacral joint and separate the sacrum from the lumbar spine. You can lift the spine with a forceps and then cut through the muscles and ribs at the left and the right side in cranial direction until the base of the skull. And then you basically make a cut in the spine at the atlanto-occipital joint. So basically, like, yeah, the most like cranial point you can get with your scissor. And yeah, so then you just like basically remove all the organs that are still attached by tissue to the spinal column, and place the spine in a 50 ml Falcon tube filled with HBSS and put it on ice. So the spinal column is now still covered in surrounding muscles and tissue, and this video shall demonstrate the careful cleaning procedure of the spine from muscle and fatty tissue. So to make the vertebral column better accessible and give you a better overview about the anatomy, you can use like a fine bladed scissor and carefully cut away all surrounding muscle, fat, soft tissue. Be careful not to damage the vertebral column, like really like sometimes also it's again like good to hold it maybe in your fingers to really feel it better in the beginning, so you don't cut into it, but really just cut the tissue surrounding it. And yeah, so as better as you clean it in this process, the easier it is in the next step. So you can, I think, nicely appreciate in this video that you can already much better see like the little vertebrae and yeah, the anatomy of the spinal column. So yeah, I think in this step it's also just important for later that like really you cut very like the ribs, like basically also, because like you will lay the spinal column on the sides, and if there are still little ribs, it will turn a little bit into one or the other direction, so it will be harder for the picking of the DRG. So yeah, once the spinal cord is cleaned from the surrounding tissue, we now have to open the vertebral column to remove the spinal cord and then also gain access to the DRG. So therefore we make a sagittal cut through the spinal column. I just here show a little bit again because the nomenclature might not be so common for everyone. So sagittal is basically this way, or here you can see it as well. And yeah, so therefore again we take a very small micro dissection scissor and put one scissor tip into the caudal spinal canal. And then you carefully cut in the sagittal plane one vertebra at a time in cranial direction until the vertebral column is half opened. The easiest way to hold the vertebral column with your fingers is with your fingers. So you have the best control over where you cut, and make really sure, or like try to be really in the middle of the vertebrae, as this will allow you the best access to the DRG later. And then you turn the vertebral column and cut through the opposite side, again from caudal to cranial, one processus spinosus vertebrae at a time in the sagittal direction. Now, yeah, the vertebral column is split in half along the sagittal plane, and you can take like a petri dish as on the image and lay both half pieces in ice cold HBSS. Then you take one tweezer and hold the vertebral column down, and one tweezer to grasp the cranial part of the spinal cord, and then pull it from cranial to caudal. Do this with both half pieces, and then the only other tip is really be careful not to, like, if you feel that there's resistance, don't pull too hard, because also, I mean, attached kind of to the spinal cord are also the roots, like the ventral and the dorsal root, and on the dorsal root you have the ganglia. So if you pull too hard, and maybe sometimes you can feel a little resistance, you can pull the DRGs already out. So if you are not sure, better go to the dissection microscope one step later, and then basically do it with a better view on what you're actually pulling out, if it does not come off easily, because spinal cord should come off easy. Yeah, and then this I just show here to better orientate yourself, again, for anatomy purposes. So once you basically lay the spinal cord on the sides, the dorsal root ganglia will be located on the bottom of this, I would call it like a half pipe. So this is also where you are looking for them under the dissection microscope. So they are in this little intervertebral foramen. And this is how it looks under the dissection microscope. So you can appreciate, like, this is like a half pipe, and really on the bottom of this half pipe, you will find the DRGs between the between always two vertebrae. And so yeah, you basically pin down one half at a time, and yeah, try to find these DRG. Sometimes it helps to locate also the dorsal roots, because they will lead you to the DRG. So you can look for them and follow them to the DRGs. And you can see that there's like a fine shiny layer over it, which are the meninges. They are sometimes pretty rough, especially also when the mice get older, like the dura mater. You can still, like, it is like harder sometimes, especially also in the more cervical area. It gets like the dura gets more like a challenge to grab through and to see also the DRGs through it. And so you basically take your tweezers, and like you can also, if you don't see like exactly the foramen, you can also try to feel it with your tweezer by basically just putting on the, like, get on the bone and feel like it's hard, and then like between two bony vertebrae you can feel that there's a hole, and there should be the DRG. So then you take the tweezer and push with slightly open ends into the foramen, and take the DRG between the tweezer endings by tightly grasping mostly like the dorsal and the ventral roots, not directly the ganglion itself, because then you damage it. And then you pull it a few millimeters by still maintaining the tight grasp, and then take a spring scissor that you have in your other hand and cut away the spinal roots and the spinal nerves that are attached to the DRG. And what you can see if you like do this, maybe on the picture is not optimal, but the DRGs are a slightly darker yellow beige in color than the spinal roots that are more whitish. I sometimes compare it with, like, if someone is also doing research with lymph nodes, it looks a little bit like a lymph node that is also a little bit more beige, and not like the fatty tissue surrounding lymph nodes, sometimes that are more whitish as well. So yeah, once you have the DRG in your tweezer, you harvest it in a 15 ml tube containing supplemented DMAM on ice, and proceed through the whole 2.5 spinal columns, or only collect the DRGs of your interest if you, for example, just look for the lumbar DRG. If you want to perform microscopy, you can also directly at this point harvest the DRGs again into 4% PFA and PBS, and then follow your preparation protocols from there. For digest and culture, once you collected the DRGs, you can, like, you spin down the DRGs at 1000 RPM, and then carefully remove the DMAM that you harvested them in, and using a serological pipette. So really, this is very important. Never use a vacuum for this, because the DRGs, they come off very, very easily, very quick. You cannot even pull the vacuum away in the time that you recognize something is coming off. They will all be gone. Like, it's very quick. So always use a pipette for this. And yeah, so once you got rid of the DMEM, you top the DRG with 3 ml of the digestion buffer, which is collagenase dispace. and incubate them shaking at 37 Celsius. The shaking is important for the dissociation of the capsule and to separate the sensory neuron cell bodies from the tissue. And here I show a representative, a tabletop incubator that can be used for this purpose. This is basically what we have here, or like something that we have here, or like it's pretty similar. And if you have like a shaker like this that rotates horizontally, similar to a Western blot shaker, I recommend to place the 15 ml tubes flat on the surface in a slight angle, as I show here, and then just fix it with tape. So then the time of the incubation at 37, that can vary. So you need 15 to 30 minutes to get a proper digest. In our laboratory we get like pretty great results with something between 60 and 90 minutes. So I mostly do 70. Yeah, but I would not go further than 90 minutes. Like something between this is good. And after the incubation, like in the digest, you spin down again the dissociated DRG for 5 minutes at 1000 RPM, and again carefully with a pipette remove the enzyme mix. And you wash them once with DMEM, and again spin them down, again carefully remove the media and replace it with the neurobasal media. And to further dissociate the sensory neuron cell bodies from the tissue, you can triturate them. And it's probably quite similar, like in the video from Evan. We just don't use like the glass pipettes that you can modify with fire. We decided to go with 3 different sizes of needles. So I take like a 1 ml syringe and start with the 18 gauge needles, which is the biggest one, and aspirate and eject the DRG neuron solution up and down 7 times, and then change to the 23 gauge needle, repeat again, aspirating, ejecting 7 times, and then continue with a 27 gauge needle, again 7 times. And then you should have like a nice solution with single sensory neuron cell bodies, and you can count the sensory neurons in a Neubauer hematocytometer to get your sensory neuron cell count that you have per sample. This is especially important, of course, if you want to like compare different mouse lines, and you need to basically adjust the cell number. Usually you should get around 50,000 to 70,000 neurons per mouse if you pick all DRGs from cervical to lumbar on both sides, which should be around 30 DRGs that you pick. For neuropeptide release and calcium imaging, it is sufficient if you plate around 3,000 to 5,000 sensory neurons per well. So I mostly plan with 20 wells that I can plate per 1 mouse that I harvested DRGs from. So at this point, you can remove the laminin drops from the pre-coated tissue culture plates and directly replace it with a 30 microliter drop of the DRG neuron cell suspension. And then you incubate this drop for 1 hour so the neurons can attach to the laminin. After 1 hour, you fill the wells with supplemented neurobasal media that also has the cytokines, for example, 100 microliter for a 96 well plate, and incubate overnight. Make sure you don't take too much volume because it's adherent cells and they might not get enough oxygen otherwise. Yeah, and this is how the sensory neurons look 24 hours after plating. And yes, at this point, you can use them, for example, for testing the release of neuropeptides or neurotransmitters through adding different stimulants. And therefore, we recommend to replace the media because like overnight, they release also a lot of neuropeptides and neurotransmitters. So if you would directly stimulate in this media, it might be a bit off because your background would already just be so high from the overnight. So replace the media directly with media that already contains your concentration of the stimulant that you want to use. And then you incubate for 10 to 60 minutes. This depends a little bit on the stimulus. usually like the neuropeptide release is very, very quick in a few minutes, but we would anyway recommend doing a time and a dose titration for every stimulant that you are planning to use. And yeah, so after the stimulation, directly take off your supernatant and freeze it on dry ice or in a -80, and then you can later use it for either ELISA or mass spectrometry to detect the neuropeptides of choice. So yeah, the protocol I was just presenting was used for multiple applications and experiments that contributed to a paper from our group from last year, where we found sensory neurons to be the first sensors of allergens in the skin. Thereby, sensory neurons get directly activated by allergens, leading to an itch response notion by scratching behavior, which is directly accompanied with the release of the neuropeptide substance P in the skin area where the stimulus basically was sensed. So the nearby Th2 skewing dendritic cells that are very close to the sensory neurons, they get activated by the substance P and then directly migrate to the draining lymph node, where they initiate the antigen presentation and the Th2 differentiation. And there is a fantastic open box science talk about this research in more detail presented by my PI, Caroline Sokol, that you can find also in the open box science YouTube channel. Briefly, by like stimulating primary sensory neurons that I prepared with the protocol I was just presenting, we performed calcium imaging and were able to detect an immediate calcium response in neurons that we challenged with our model allergen papain. And as you can see, like by basically doing a sequential stimulation with histamine, chloroquine, capsaicin, and potassium chloride, we were able to classify basically the neurons that respond to our allergens. And we also assessed neuropeptide release through allergen stimulation, once by using a dermal explant in vivo model, where we injected the allergen intradermally into the skin, followed by a punch biopsy and ELISA for also substance P and CGRP. And we then confirmed these findings by using the protocol I was just presenting, and with like these commercially available ELISA kits, again looking for substance P release and CGRP release, we could see that papain, our allergen, is inducing substance P release, while it is actually decreasing CGRP release in vivo and in vitro. So yeah, I hope with this I could offer some insights into possible applications for the DRG dissection protocol. And with this, I want to say thank you to Carrie, who is an amazing mentor and PI, as well as my lab members and our collaborators from the Leibniz Institute in Jena and the Harvard Medical School in Boston. Thank you for your attention.
It's fantastic. Thank you, Caroline. I have never done DRG coaches, but this is very helpful for me. So it's time for questions. I want to remind everyone that editor Mary Sun is still with us. So if you have a question about STAR protocols, you can also take this opportunity to interact with her. We have one question in the chat. It's regarding the purity of the DRG cultures. So what % of the total cells in the culture are neurons? And have you characterized the contaminated cells, like glial cells, fibroblasts, or other myeloid cells in your culture?
Yes, so it's a good question. There are, I think like most of the cells that I see also like in culture are probably also glial cells, that you can a little bit, I mean, for the culture, like in the cytokine mix, you can use RRC, which will a little bit more disturb the growing of the glial cells than the ones of the neurons. On the other hand, I mean, RRC is also, if you, depending on what downstream applications you want, it might also influence your health of the neurons. So this is definitely an issue. I think like the other cells, honestly, because it's a very long digest that we are using, the few immune cells that are in, I think they don't make it through the 90 minutes or 70 minutes collagenase dispace digest. So I would not too much worry about these. We indeed have a protocol like for basically looking for immune cells within the DRG, where we don't do the digest, but directly homogenize the DRGs as they are, and then also do like a max, you can do a max isolation of CD45 cells and from this solution, but we like exactly to get more healthy immune cells that were in the DRGs. But yeah, we haven't like further analyzed these the cells, but yeah, in the dish you can see mostly I think glial cells.
Okay, thank you. And I'm just wondering, how long can you culture the DRG cells in the incubator?
We mostly just do for 24
hours?
hours because
Okay.
mostly we do either the calcium imaging the next day or then directly the neuropeptide release assay, where you just do a quick stimulation. And then we have also tried already for like longer days, because now we have some experiments where we basically do a challenge with the stimulus and then we do a challenge again another day and look a little bit for like changes there. So you can definitely grow them for I would say 72 hours. They are still like happy if you change the media and give them like new cytokines again. And like yeah, don't put, as I said, too much media on top, because they don't breathe then anymore that well.
Okay, thank you so much. Yeah, so I think we are running out of time. No more questions. Thank you again with Evan and Carolyn. So both of them are on the transition to become independent PIs. So please check out their work and the openings in their future labs, maybe. And also thank you, Editor Sun. Thank you for sharing STAR protocols, benefits, and other information with us. Yeah. So this is our last seminar, Open Box seminar in 2021. We already have exciting seminars aligned in the coming January and February. So please keep following us on social media to learn new updates. Thank you again and happy holidays. I'll see you next year.
Thank you.
Thank you. Bye-bye.
Thank you, everyone.